Hello, my name is Finn Burrelson. Today I'll be discussing RMSX and Flipbook, which are tools that let you see when and where proteins move during MD simulations. Let's take a look at an MD you may run into in the wild. So, the way MD simulations work is you start with a static picture, just like a PDB, of where all the atoms are in space. And then you use a complex program like NAMD or GROMACS or others, which is able to take that frame and predict from one time step to the next what forces each atom feels and how those atoms are going to move between one time step and the next. By understanding how these proteins move and change shape, we can understand their function better. This could be how they're involved in diseases, it could be how it performs their functions, it could be how information is transferred. Let's think through a protein simulation, but before we deal with 3D, let's imagine a short peptide sequence moving in two-dimensional space. We'll use this 11 residue peptide. So, our chain moves over time under the forces, some get closer, some get further away, and we can make this a little animation. But we might want to know, how can we quantify this movement? To start, let's just see what this entire simulation looks like laid over top of one another. From this, we can see that, for example, position 6 didn't move very much, but position 8 did. What we need, though, is some sort of measurement of how much things are moving. How can we actually compare position 6 versus position 8? There already exists a handy tool that has existed for some time for understanding MD simulations, which we call RMSF, or the root mean squared fluctuation. What it does is it quantifies how much movement there is at a single atomic position over the course of the simulation. So for example, for position 6, how much it fluctuates around in space. This can be useful for measuring how dynamic a position is, but it also is helpful for isolating areas that are likely to be more flexible. While RMSF is useful for telling us about particular positions, which one is more or less dynamic, what it can't tell us is how it changes over time. If we want to understand how the complex as a whole changes, we can use something called root mean squared deviation. RMSD operates a little bit differently than RMSF. In RMSD, you need to have a template. Oftentimes, we use the very first frame of our simulation as a template. We can treat the very first frame of the simulation as a reference structure and use it to compare and measure the difference to subsequent frames. Let's try an example. We'll superimpose the second frame on top of the first frame, and we'll use it as a reference, and we'll measure how much between the first frame and the second frame each position is changed. This average deviation across all of the atoms is what we call the RMSD value. What we end up with is a timeline of comparisons, many comparisons to this reference structure, which tells us over time how different each frame is from its reference structure. A high RMSD value means it's more different from that reference structure. Let's recap these two methods. First, RMSD. RMSD tells us the average spatial deviation of the system over time by averaging the deviations of each atom to the reference. On the other hand, RMSF displays the average fluctuation of each residue during the simulation. So, RMSD quantifies change over time. It tells you when things have changed more. On the other hand, RMSF tells you the movement per residue. Basically, which residue moves more. Now, both of these metrics are quite useful, but it can be difficult to get a complete picture just from looking at these two. If we see, for example, that there are spikes near the end of the simulation in the RMSD plot, then we might know that that time period is pretty different from the reference frame. And if we look at the RMSF plot, we can see that a lot of the motion seems to be concentrated around position 50. But what we don't know is that spike near the end of that simulation the result of shifts around position 50, or is it 25 and 75? We don't know what is shifting when, and what is accounting for the spikes on one versus the other. Now, what we'd really like is some kind of metric that can capture both change over time and per residue information. Some kind of metric that can fall between these two. Perhaps some kind of heat map. How we can actually accomplish this is pretty straightforward. We can just break our simulation into smaller pieces. For example, the first third, the middle third, or the final third. But we could also repeat this RMSF calculation over many, many slices. And if we do, we might end up with something that looks like this, with a surface. And the surface can be represented as a heat map. Now, the reason this heat map is so useful is because now we can pinpoint what is moving, where it's moving, and how much it's moving, which is not something that we could get from either the RMSF or the RMSD plots alone. Now, this heat map representation of the entire protein chain can tell us some very useful information, but in terms of getting an intuition for what this actually means in 3D space, most of us probably don't have the entire structure memorized in terms of what position corresponds to what feature inside of their protein chain. And so it would be helpful if we had some kind of molecular representation that could go with this, that could help us see exactly what these highlighted areas represent biologically. Let's talk through how we could do this. First, we could take the first time window. Let's take some snapshot from window 1, maybe the last frame within this time window. Using the RMSX values we generate in our heat map, we can map those values onto each atomic position, and we can scale and color them to make it easier to see. You can see that position 50 is the protease's active site, which is useful information. Now, you just repeat this for each window, and we end up with this nice picture of how the structure changes over time, mapped onto its three-dimensional structure. So we can see areas in which the chain moves more, it moves less, and how it goes through time. This approach to coloring and scaling protein structures resembles those little children's books where you can flip through the images to get a sense of how things move and change over time. And so we call this method Flipbook. Some people may wonder, well, how can I apply this method to something that has multiple chains in it? Well, all you have to do is plot them separately and then combine them. We can automatically sync these together so that we can create multiple plots, one for each chain. For example, in this protease example, chain A and chain B. We can also create flipbooks either for each of the individual chains or a larger flipbook that contains all the chains with all of their information written to them. Now, I mentioned that this method requires selecting a time window. What if my simulation is very long, or if I want to use a very fine window? Let's look again at a longer simulation and then focus in on two areas of interest. There are periods of high stability and periods of low stability here. I'll underline the low stability areas in blue for both chain A and chain B, and the areas of low stability in red. Let's focus in on this area by just looking at the flipbooks from this region of high stability. You can see that the active site largely remains shut within our snapshots, and the RMSX values are pretty comparable to the rest of the structure. There's some minor fluctuation, but it seems to be from these snapshots that they are sticking together, and because of the low RMSX values, we know that they're staying together, otherwise the fluctuation would be higher. On the other hand, if we look at this period of low stability with high RMSX values, we can see a very different story around the active site. We see high RMSX values, and we can see from the snapshots also that they're changing conformation a great deal. They aren't remaining in one single state, but sort of flapping open rather than settling together. If you're interested in trying these out for yourself, I'll just give you a very quick preview of how it works. This is all built to work inside Jupyter notebooks. From it, you just need to take a PDB file and your trajectory file. It supports most major simulation suites, so NAMD, GROMACS, LAMPS, etc. And what it produces within your notebook is publication-ready plots, exactly as you've seen here. You can also create the flipbooks directly within the notebook as well. It will actually launch in your preferred viewer, whether that's Chimera X or VMD. And from that, you can tweak, rotate, and adjust it to make the visualization that you need. A quick plug for our package. It's built on MD analysis, which allows for very quick data manipulations. We include Jupyter notebooks that have everything you need to run them. You can use your own data as well, simply plug in and replace the paths. It's designed to get plots with minimal effort and have comprehensive documentation. All you need to do is go to our GitHub and download it to try it out for yourself. To give a couple more concrete examples of how these tools can be used, say you study something like allosteric changes, where you may have something binding to one side of the protein affecting the shape or the function of another side of the protein. Both of these tools, RMSX and Flipbook, can show how things are propagating through your system. On the other hand, if you have SMD simulations, let's say, right, these are systems, steered molecular dynamics, where you're in some way affecting your system through external forces. You can see, for example, how pulling occurs, how force and motion propagates through your system. You can see what moves first, what moves second, and what moves finally. You can compare this with things like dynamic network analysis to get a more complete picture of how these proteins are responding under force. Finally, just thinking about Flipbook as a tool unto itself, you don't have to run it with RMSX. It actually works with a number of different other metrics. For example, trajectory maps, that's just the displacement from a reference frame. You can look at hydrogen bonds, and we recently also included the local distance test, lLDT. You can pretty much use any per residue metric that you would like, whether that's window-based like RMSF or per frame-based like RMSD. really any of these things can be implemented, and we provide the tools to do so. Thank you so much for listening. This has been an overview of MD simulations, talking about RMSD and RMSF, and finally introducing our new tool, RMSX and Flipbook. If you have any features you'd like to request or any metrics you'd like us to add to the system, we are happy to do so, and we've already done so with lLDT. In the meantime, I hope that you will try this tool out yourself. All of the links can be found in the description below. We encourage you to try it out, and again, if you have any issues, questions, comments, or concerns, please feel free to reach out. Thanks again for checking us out, and I hope this tool is useful for you in your research.